everyone. I thought it might be helpful to have a video resource for our final exam cumulative stuff preparation. So that's what this time together is going to be on. What are the big picture things that are on the final? What are some of the key concepts that you should be reviewing? What's the level at which you should do the review? My perspective with the final exam is we don't need to get bogged down in a lot of details now because quite honestly, between your quizzes, your learning modules, the problem-based learning, and all those exams you had, we've had plenty of assessment of details. I've done a lot of checking on your level of detail. So with this cumulative stuff at the final, at the end of our semester, it really is about, for me, big picture, take home, overarching things and themes in our course. So that's what we're going to spend some time on here. At the beginning of the semester, the first organ system we emphasized wasn't an organ system at all. It was actually a connective tissue, and that was blood. So we learned about the formed elements of blood and the plasma. In the lab, we looked at clinical tests. In the lecture, we talked about its regulation and what it carries and all of its functions and all of that stuff, right? So that's that detail. So what we're going to look at now is what's on that study guide for the final exam and what kind of stuff can you expect to see? What should you be ready for? So here's one of those processes. We had to learn where do the formed elements even come from and how do we control those processes? Well, where they come from and how we control that is hematopoiesis. And it consisted of erythropoiesis for making erythrocytes, leukopoiesis for making leukocytes. The third one we looked at back in the day, that was thrombopoiesis, and that was for making thrombocytes or platelets. But here at the end of the semester, let's focus and zero in on these two that you see here. So erythropoiesis. Again, the end result and the purpose is to make red blood cells, make erythrocytes. Well, one of the triggers for how many erythrocytes are we going to need to make right now is something called tissue hypoxia. As blood travels through your kidneys, oxygen levels are monitored. And when oxygen levels are low or tissue hypoxia has occurred, cells respond to that in the kidney by secreting erythropoietin or EPO. And EPO will travel through your blood supply until it ends up at your red bone marrow, and it acts on the stem cell and triggers that stem cell and tells it to go down a developmental process and become an erythrocyte for us. Well, the end result would be a rise in the percent of packed erythrocytes or a rise in your hematocrit. Now, all those details in between, the different cell stages during that developmental process, the tissues and cells in your kidney, all of that stuff, that's a bit detailed for me for a lecture final exam. I only have so much time and so many questions to ask you about that old stuff. So review what you see in front of you, that erythropoiesis is the process for making erythrocytes. That the trigger in this homeostatic loop is tissue hypoxia. The efferent pathway is erythropoietin. The target is going to be the hemocytoblast in your red bone marrow, and the end result of that is a rise in hematocrit. Also familiarize yourself with the fact that this is a negative feedback loop and what that means. Now, why do you see some pictures? Well, once upon a time we talked about, like, what are you going to need to be able to do all this? What are the resources your body's going to need? We talked about a lot of resources back in the day. So for now, familiarize yourself with the fact that, well, you're going to need some iron because you need the iron to make the hemoglobin for these millions of erythrocytes. You're going to need vitamin B12 to help drive this process. And even if you eat vitamin B12, you're going to need your stomach to give you some intrinsic factor so that you can absorb that B12 in your intestines. And one of the other processes that falls under the hematopoiesis umbrella is leukopoiesis, which is how we make leukocytes. this one's a bit weird. And once upon a time in class, I was like, what came first, the chicken or the egg? What came first, a leukocyte or the leukocyte? And that's kind of how this process works. So you're already existing and you've already got a leukocyte population and you undergo an immune event. So maybe bacterial or viral infection or something else. And your leukocytes trigger an immune response. And when we learned that immune response, we learned that there are lymphocytes that will secrete very specific colony-stimulating factors and interleukins. And some of those go to the same stem cell, same stem cell in the red bone marrow, and trigger that stem cell to become a very specific leukocyte. So that's the level of hematopoiesis you can expect to see on the final exam. Now, formed elements. I'm not going to show you a photomicrograph on the lab exam, from a lab exam on your final and ask you to identify a formed element. But just in case there's a description of one of them, you should know who we're talking about here. So, no granules, large round nucleus, lymphocytes. No granules, C-shaped nucleus, monocyte. Lilac granules and multilobe nucleus, neutrophil. Red granules, eosinophil. Large purple granules, basophil. Red and biconcave, erythrocyte. Very small, platelet or thrombocyte. Now you also need to be able to describe general functions of each. So erythrocytes, gas exchange, carry respiratory gases, things like that. Platelets, clotting and coagulation. Monocyte or macrophage, they're phagocytic, but in the long term. Lymphocytes, oh gosh, there's so many of them, right? So antibody production, immune response, helper T cells, cell-mediated response, that all falls under the lymphocyte category. Eosinophils were allergic responses and parasitic infections, microscopic parasitic infections. Basophils, inflammation with histamine and heparin. And neutrophils are also phagocytic, but they're short-term. So let's get into some of the processes. You should be able to take someone outside the class and at a fundamental level explain the process of hemostasis, put the clotting factors in their correct sequential order, and describe some of the nutrients that are required as part of the coagulation clotting process. So let's go over those briefly. Now at the top in the green arrow there, you have the steps of hemostasis right there. So those are the steps of hemostasis, okay? So the first thing that happens when we damage a blood vessel is the smooth muscle in the tunica media contracts. And that's a positive feedback loop, and that closes off the broken end of the blood vessel. Now as vascular spasm is going on, any platelets that are coming by and encountering that damaged blood vessel wall will begin to stick to the damaged wall and to each other and literally form a plug, and that's called a platelet plug. So the second step is platelet plug formation. Now, once the platelets start to stick to each other, they'll start to release their cytoplasmic granules. Once upon a time, we talked about what's in those granules, but that's too detailed for a final exam question. Once the granules are released and they become activated, they will trigger the coagulation cascade of chemical reactions. To be able to put those events in order and to explain what's happening in each event. Now, once we activate that cascade, earlier in the semester we talked about steps in an intrinsic pathway versus steps in an extrinsic pathway. So you don't need to know the details of those two pathways for the final exam. But both pathways get us to the same endpoint with clotting factors that you need to be able to put in order. And so both pathways get us to prothrombin activator. And once you've created that molecule, it's going to act like an enzyme. So prothrombin activator will take prothrombin, already floating in the plasma but inactive, and convert it into thrombin, which is an active molecule. Thrombin will kind of act like an enzyme now. And thrombin will take fibrinogen, a soluble, inactivated molecule floating around in the plasma already, and convert it into fibrin. Fibrin is insoluble. Fibrin will be those fibers that come out of solution that we once saw on an animation in class together. And then that's what the clot will be made of. When the fibrin proteins come out of solution and stick to each other, and the walls or the edges of the broken blood vessel and the platelet plug, they'll just kind of trap everything in place, and that's what a clot is made of. So again, be able to describe the 3 steps of hemostasis, put them in order, and put the clotting factors in their correct chronologic order. Now, in the lab earlier in our semester, we learned how to type blood, about antigens, antibody production, hemolytic reactions, and we learned how to manage a blood type. And that was lab. In lecture, I did mention blood types early on and the importance of compatibility. So you are going to get a random blood type on your lecture final exam. And you need to learn how to manage that blood type or explain how to manage that blood type. So in other words, blood type WZ. So you need to be able to take blood type WZ and be able to predict what are some of the blood types that could be donated to, what are blood types it could receive from. So I'm not going to go through all of them, that's your job now, but one of the things we did earlier this semester was use the Nobel Prize blood typing game for practice in blood typing. So if you struggled with blood typing way back, definitely check out the resources on Blackboard for blood type. And again, you will get a random blood type on the lecture final exam and need to be able to determine donate to, receive from. If you have questions, you can get back to me on that. So that's the kind of stuff to review intensively for the lecture final exam pertaining to blood. So now let's move on to heart. What kind of stuff are you responsible for on your cumulative final exam? So here's some stuff. So layers of the heart wall. From the outside in, right, we had an epicardium, which is also called a visceral pericardium. That's simple squamous. Then you have the middle part that's made of cardiac muscle tissue. That's the myocardium. And you have the innermost layer that's also simple squamous tissue. That's the endocardium. Now, epicardium and endocardium kind of have the same function. They reduce friction. They do it in different ways, but that's their job. The myocardium, since that's where the muscle is, that's the contractile stuff. So that's what's responsible for making the pressure changes of the cardiac cycle. Now on that photomicrograph on the right side of your screen, you can see the cardiac muscle tissue. Because the other part of this is, well, what's unique about histology of cardiac muscle cells? And there are several unique things. So they are striated. They are branched or Y-shaped, which means there's a little connective tissue between them, here and there. They're under autonomic nervous system control. And the big thing about them, they have intercalated discs. Now, intercalated discs allow these cells to have a flexible connection where they share cytoplasm. So when they release calcium, all the cells are going to share the calcium in the cytoplasm. So they're going to act as a unit. In lab, we said that's a functional syncytium. Okay? And so the importance and the relevance of intercalated discs in cardiac muscle cells is that it allows the interconnected cells within a bundle to act as a functional unit. That's what's unique about the heart with histology and layers of the wall. We also talked about what's unique about the heart with its physiology. Remember, the heart has its own nervous system. It has a pacemaker, that's a ganglia that sets action potential rhythm, like fire an event, fire an event, fire an event. And its nickname is a pacemaker, so I'll point it out on the diagram in a second. Now those electrical events are going to leave that pacemaker and they go to a second set of ganglia that delay the signal so that atria and ventricle eventually are out of phase. So again, we're going to have a pacemaker. It sets the rate. And then we have a structure nicknamed the gatekeeper that delays the signal. Now, after that, those action potentials would move down the septum, and then they kind of separate out from each other at a structure. And then we have the equivalent of motor neurons going through ventricle myocardium. So who's where? Well, SA node, that's the pacemaker. AV node, that's the gatekeeper, that's the delay. And then you had, it's not written on there, you had a bundle of His, that's the separation point. And then we have all these Purkinje fibers, the motor neurons. So that's the kind of electrical conduction system of the heart. But we also learned about how the electrical events are different. So in the SA node, we have things called pacemaker potentials. And back in time, we kind of compared and contrasted those to an action potential. So let's just look at what's unique about pacemaker potentials. And there's several things. They're in the SA node. The other thing is magic numbers, okay? So they're at rest at around -60. They depolarize at around -40 millivolts. And it's sodium that gets them there, gets them to their threshold. But once they hit threshold, it is calcium that moves. So at threshold, they will open a calcium voltage-gated channel. Calcium flows in. And because we have a different ion, we have different magic numbers. We have different millivolts. So at threshold, calcium gets us from threshold, and then its peak is just above 0 millivolts. So it's a much shorter amplitude. So by using calcium, the SA node cells are basically causing a really short electrical event. So right about here, we hit peak, and then we open potassium-gated channels, and we repolarize. So that's a few things that are unique. Okay? Again, we have different numbers. We're using calcium to depolarize to peak. The other thing that's different here is they don't really hyperpolarize, right? A regular action potential gets like extra negative. Pacemaker potentials don't get extra negative. So with a pacemaker potential, again, we're sitting at rest. Calcium flows in, you depolarize with calcium. What did I say? You're sitting at rest, sodium flows in. We will open a calcium voltage-gated channel at threshold and hit a peak, but the amplitude is a little. Then we'll open potassium, go right back to threshold, or right back to rest, and we're close to threshold. We're ready to do it again. Okay? So again, what's unique about pacemaker potentials? The voltages are different because we use a slightly different ion. Okay? So sodium gets them from rest to threshold. Calcium gets them from threshold to peak. Potassium gets them from peak back to rest. No hyperpolarization. So that's what's unique about that pacemaker potential. Now, we did use magic words earlier on. We were like, because you are basically rest, threshold, peak, back to rest, they will hit threshold very easily and automatically, and we say they self-depolarize. So you want to be able to, again, briefly explain SA node, AV node, bundle of His, Purkinje fiber function, put them in the correct sequence of events. You want to be able to explain a little bit about pacemaker potentials, why they're a little different, what calcium is doing for us in this electrical event. Now, once upon a time I said, wouldn't it be cool if we could watch these electrical events moving through the heart structures? And we can. And when we monitor them and we watch them move, that's an EKG. So you want to be able, with an EKG, to recognize and explain, big picture, electrical events and mechanical events. So this slide has the electrical events: P wave, QRS complex, T wave. Electrical events are things like depolarization, repolarization. So P wave, atrial depolarization. Action potentials are arriving and moving through. QRS complex, ventricular depolarization. Action potentials are arriving and moving through the ventricles. T wave, that's ventricular repolarization, and you can think of that as like the action potentials going away. So those are all electrical events. Mechanical events are a different story. Mechanical events are things like systole, diastole. Mechanical events are not directly seen on our EKG. They are not the waves. They're actually the flat line after a wave. So the mechanical events follow the electrical events. So atrial depolarization, that's an electrical event. In the flat line that follows, the atria myocardium should contract or go into systole. QRS complex, well, that's an electrical event. That's ventricular depolarization. In the flat line that follows, the ventricle should contract or go into systole. T wave, ventricular repolarization. But in the flat line that follows, the ventricle should relax or go into diastole. The only thing we really can't see on the EKG is when does the atria repolarize, and that's during the QRS complex. So again, you should be able to explain each of these three deflection waves we went over, the electrical event that we're actually measuring, and what mechanical event should be following that wave. Okay, so far with heart, right? We've looked at its layers, the tissues they're made of, their function. We looked at its histology, intercalated discs. We looked at its mini electrical system, SA node, AV node, bundle of Purkinje fibers. We have looked at its pacemaker potentials of the SA node, things like calcium, different millivolt at peak, no hyperpolarization, self-depolarized. So now, we've even watched pacemaker potentials start and action potentials move with an EKG and looked at deflection waves and what we're measuring. So now we can zero in on cardiac cycle. Cardiac cycle is the story of pressure changes and how those pressure changes open and close valves and move blood through the chambers of the heart. So, contracting. When a chamber contracts, it's because action potentials just told the muscle to contract. But when a chamber contracts, imagine that chamber squeezing, and it's squeezing on the blood inside of it. So the pressure that we're going to generate is going to go up. The harder you squeeze, the more pressure you will make. Conversely, when a chamber relaxes, that's because the action potentials went away. And so the less you squeeze, the lower that pressure becomes. So the mechanical events are contract and squeeze and raise the pressure versus relax, stop squeezing, and lower the pressure. And because of the way the SA and AV node works, we're going to go back and forth between those two states, squeezing and generating pressure versus relaxing and dropping pressure. So the best way to kind of focus in on that is to look at ventricular systole. You know, the atria are kind of, don't tell them I said this, the atria have thin walls and they're kind of a little bit weak, like me right now. Whereas the ventricles are like the bodybuilders of the heart, right? So the ventricles really are going to drive the process. So ventricular systole, action potentials arrive in the ventricle, ventricle begins to squeeze and pressure begins to rise. The first thing that will happen is we will close the respective AV valve. Now the ventricle will continue to squeeze and pressure will continue to rise. And eventually we will generate enough pressure to where we will push open the respective semilunar valve. And that's when the blood ejects. Then the action potentials go away and the ventricle begins to relax, so pressure begins to drop. The first thing we will do is close the semilunar valve. The ventricle will continue to relax and pressure will continue to drop. And eventually the pressure will become so low in that ventricle that we will pop open the AV valve and blood will enter. So be able to explain that. I'm going to go through them again and write like in notes, the atria is the opposite. And that'll be good enough, okay? So again, action potentials arrive, ventricle begins to squeeze, pressure begins to rise. Close AV valve, pressure continues to rise. Open semilunar valve, blood ejects. Action potentials go away, ventricle begins to relax. Semilunar valve closes, ventricle continues to relax and pressure continues to drop. Eventually, pop open AV valve and blood enters. So that's basically the cardiac cycle. The last thing I believe that's on your bullet point list, that study guide you should be filling out, has to do with circuitry and tracing. Now I'm not going to say trace a drop of blood from this to that. I've done that already more than once. What I'm talking about here with tracing blood through is the circuits, right? So the vena cava, right atria, tricuspid valve, right ventricle, pulmonary semilunar valve, pulmonary trunk, pulmonary arteries, even pulmonary veins. They're pulmonary circuit. They're all about getting blood to and from the lungs for gas exchange. The entire rest of the circuitry in your body all belongs to systemic circuit. So like left atrium, bicuspid valve, left ventricle, aortic semilunar, aorta, and all the arteries you learned names of earlier this semester, they're all considered systemic circuit. So when that bullet point says, can you trace, I mean in terms of like which vessels belong to which circuit and not something like we did in lab. So after we were done with heart, it was early in the semester still, we were young, we went on to blood vessels and blood pressure. So let's take a look at those kind of bullet point lists on your study guide for that stuff. So one of the things on the study guide, it says, well, what are the 3 tunics of blood vessels? And what are differences between arteries, veins, and capillaries? So here we go. From the outside in, outermost is tunica externa or tunica adventitia. It is primarily connective tissue. It is where the nerve endings kind of come in and are housed in there, those nerve endings that will control the muscle. Middle is tunica media. Tunica media is where the smooth muscle is. So the function of tunica media is constriction and dilation. Outer or innermost is tunica intima. Tunica intima is also called endothelium as a layer. Tunica intima is simple squamous epithelia. It reduces friction. Now in the differences between the different types, arteries have the thickest tunica media. Veins have a thicker tunica adventitia. When it comes to capillaries, they're the exception to the rule. Capillaries have exactly 1 tunic, and that's it. They have got the tunica intima, and we just call it an endothelium. So be able to, again, explain, identify those 3 tunic types, their tissues, and a general function, and compare and contrast at a basic level arteries, veins, and capillaries. Now, we even went into different kinds of arteries, different types of veins, and different types of capillaries. And we've seen these 3 capillaries now throughout the semester. So familiarize yourself with their terminology and their functionality. So continuous, right? Continuous capillaries. They are for small objects. They're the clefts between their cells are small and not that frequent. So small things, small nutrients, monomers, gases, things like that cross their endothelium. Fenestrated capillaries, lots and lots and lots and lots of holes. Fenestrated capillaries, so like an example is a glomerulus. So with fenestrated capillaries, you get small molecules, but bulk fluid movement, which means fancy way of saying lots of water at once, which is exactly what happens when we filter blood at the glomerulus. So fenestrated capillaries, bulk movement of fluid. An example is a glomerulus infiltration in a kidney. Now the last one here, sinusoidal. Even if you look at this diagram, you can see they have the biggest, most, we'll just say biggest clefts between, the biggest spaces between the cells. So they are for big things. So what crosses their membranes are things like plasma proteins, immunoglobulin cells. So we'll find them more in like liver, bone marrow, and things like that. Wherever you have to pass either proteins or cells across a membrane. So familiarize yourself with the 3 types of capillaries, example of where you would find them, and the kind of molecules that cross over each. This was the hardcore stuff. It all started fine with blood vessels, and everyone was like, oh, that's really interesting. And then we got into 3,000 mechanisms for controlling blood pressure, right? So we don't have to get too hardcore now. Again, we're at the end of the semester. What's the big picture, right? And so here's some of the big picture things. So familiarize yourself with these mechanisms and these concepts. Now, how is blood pressure regulated? 2 ways. Actually, 3 ways. To change your 120/80, to change your systemic blood pressure, you could either change your heart rate, so make blood pump faster, or you could change your blood volume. You can change how much blood you have in your tubes, right? Blood pressure in general is just the force blood places on the walls of the tube as it's sitting inside one of your tubes. That's all it is. It's just the force the blood is putting on it. And so the more blood you put in a tube, the more force you create. The other way of changing blood pressure is to change the size of the tube so it comes in contact with the blood more. So earlier in the semester, I was like, if you want to change blood pressure, you got to change one of 2 things, really. You're going to change the size of the tube, or you will change how much blood is in that tube. So changing diameter of the blood vessel, constriction, dilation, that's changing the size of the tube. That's pressure and resistance related. Changing blood volume, well, that's changing how much blood is in that tube. And so again, how do you do these? Well, again, that's a little bit more detailed. So when we talked about autoregulation and myogenic and all these like lactic acid buildup, that's an indicator of metabolic activity, and here's how the smooth, that's all too detailed for a final exam. I know, the dog is back again, y'all. And so focus on what you see here. So vasoconstriction increases blood pressure by way of resistance, and that's the job of tunica media. Vasodilation decreases blood pressure in that vessel by way of resistance. And if you want to move blood this way, you need a high pressure spot versus a low pressure spot. So if we wanted to control blood flow down to 2 capillary beds right next to each other, then this capillary bed may be high pressure. We're going to push blood out of it, and we're going to push it into this capillary here. That's low pressure. So you just need a high pressure spot and a low pressure spot, and blood will move high to low, like everything in the body. Okay. We talked about how you will do that between organ systems. So like you just ate your big holiday meal, and you've dilated the blood vessels to your digestive tract, so the blood goes into your digestive system, and that's why you're like, huh, what? Because your brain might not be getting as much blood as your ileum is at the moment, okay? So if you want to move blood between 2 points and be very specific about it, you will rely on changing diameter of the blood vessel with these 2 mechanisms. If you more have a problem with your 120/80, so we're not so much concerned about moving more blood into our skeletal muscles because we're exercising and less blood to our digestive system. We don't really want to do that. We want to stay alive. Then we change blood volume. So changing blood volume is really related to your 120/80. Can you just have some blood in your body and move it, for the love of God, right? Whereas your diameter of your blood vessel is about fine tuning, like, oh, this organ needs a little bit more right now, that needs a little bit less, we'll flip it around later. Okay, so diameter of blood vessel, fine tune control by constricting, dilating. Blood volume control is more about systemic, body-wide, 120/80. So how do we do the blood volume thing? You've got lots of options. Okay? You have atrial natriuretic peptide. That's released when your volume is too high, so it forces your kidneys to secrete sodium and water and get your volume back down. Antidiuretic hormone, the exact opposite. Blood volume's too low, or blood pressure's too low. So it tells your kidneys to reabsorb more sodium and therefore reabsorb more water and increase your blood volume. Renin is listed there. Renin is what triggers the renin-angiotensin pathway. And if we go all the way to the end of that pathway, one of the things we see at the end of that pathway is aldosterone. And aldosterone works like ADH. It tells your kidneys, reabsorb more sodium and water. So ADH, aldosterone, make your blood volume go up to restore homeostasis. ANP makes your blood volume go down to restore homeostasis. The other thing you see there is water, sodium, glucose, okay? So if you think about it, ADH, aldosterone, ANP all work the same way. They tell your kidneys to change how they are transporting sodium. And we said all semester long, where sodium goes, water follows. So if you reabsorb more sodium, you'll reabsorb more water. If you release or secrete more sodium, you'll secrete water with it. Glucose is there simply because I pointed out diabetes a lot all semester long, right? It's like glucose is kind of osmotic. Where glucose goes, water also follows. So we don't intentionally use glucose to control our blood volume, good God, no. But that's why we see the symptoms of diabetes, uncontrolled diabetes mellitus, when we start getting too much glucose in the blood. We get too much glucose in the filtrate in the kidneys, and we talked about a couple of times throughout the semester what blood glucose can do to blood volume. Okay, so again, how do we regulate blood pressure? Two ways. We can change diameter of the blood vessel with constriction or dilation. That is fine-tuned control. That allows us to have an organ system, an organ, structures within the organ or neighboring capillary beds get exactly the amount of blood they need moment to live. The other way to change our blood pressure is to change our blood volume. That's a systemic strategy. That's literally in your body, you're too dehydrated or you've lost blood and your pressure's too low to move anything properly. Or your pressure's too gosh darn high and you might have a stroke. Okay, so changing blood volume is body-wide control. Changing diameter of blood vessel, fine-tune control. That's the level at which we're going to do for final exam. Get all that time spent on diagrams and tiny little things. That was earlier in the semester. Okay, next, our organ system. After we had Simage-Fem with blood pressure, we moved on to the lymphatic and the immune systems. So again, diagrams. So with the lymphatic system, you have a few bullet points. One of your bullet points is like, well, what's a lymph node? So here's a lymph node for your viewing pleasure. More afferent vessels than efferent, so flow of lymph fluid is really, really slow through the node. Lymph nodes at the level of a final exam, they filter and process lymph, and they house the immune system. Don't go in more detail within that for the final, okay? On the left, we also see this diagram. This diagram is to remind us that these are a lot of different types of lymphatic organs, right? And so a lymphatic organ is considered lymphatic because it houses the immune system and it has shared anatomy. Some of these have lymph fluid running through them, some of them don't, right? You don't have to get hardcore about the details of it now. The other thing that's on your bullet point list for the lymphatic system is over here, kind of on this diagram, and it asks you about how does lymph move through the body. So we collect lymph in a lymphatic capillary because there's a pressure gradient. Lymph capillaries, highly permeable. They have one-way valves. Once you start to move the fluid, and we rely on skeletal muscle, right, and respiratory pump, muscle pump, respiratory pump to move lymph through them. Once you start moving the lymph through them, then it'll go again, collect it in a capillary, move it in a vessel, filter lymph in a lymph node, move it out on a vessel. Smaller vessels converge to form bigger vessels called trunks. And you will eventually return it to the blood supply in a duct, one of these two ducts. Now, if the lymph is coming from the upper right quadrant of your body, that ends up in the right lymphatic duct. Everywhere else in your body, that lymph is going to end up in the thoracic duct. So again, how does lymph move through? Well, pressure gradient drives it into the lymphatic capillary. Skeletal muscle pump and respiratory pump push it through lymphatic vessels. As it makes its way and flows through a lymph node, it'll be filtered. Large vessels converge to form trunks, and if it's the right upper quadrant of your body, that those trunks will dump into the right lymphatic duct, and you'll recycle it into your blood supply. If it's anywhere else in the body, we have one more step. So if it's left upper quadrant or anywhere else in your body, you're going to collect it in capillaries, move it in vessels, filter it in a node. Smaller vessels converge to form trunks, and then in your ventral body cavity, those trunks will converge to form something called a cisterna chyli, and the thoracic duct comes out of that. So again, how does lymph move through the body? Well, if it's upper right corner, collect it in a capillary, move it in a vessel, filter it in a node, return it on right lymphatic duct by way of trunks. If it's anywhere else in the body, collect it in a capillary, move it in the vessels, filter it in a node, collect it in trunks, gather it in a cisterna chyli, return it to the blood supply in a duct. And again, how do we move the lymph through all of this since there's no pump? Skeletal muscle activity, respiratory pump activity. And I think the last thing to remind you about is as lymph moves through lymph nodes, super duper slow. Remember I showed you I Love Lucy candy conveyor belt, right? So lymph flow, once you get it moving, super duper slow. That's it for the lymphatic system. Now with the immune system, that was, I think, everybody's least favorite organ system. What? So one of the things you want to be able to do is compare and contrast innate and adaptive, and we'll do that a little bit in a sec. And then, what are the events of the immune response? So innate, things like, here's keywords for you. Innate, functional at birth, no memory. It is primarily barriers and strategies. So all the different barriers of your body, fever, inflammation, phagocytosis, complement. Its intention is to prevent entry or prevent spread. And it's not specific at all. It doesn't matter what the pathogen is. The innate strategies are just trying to prevent entry or spread of the pathogen, but they're not designed to affect any particular kind of pathogen. Now, if that pathogen spreads, right? So now we activate adaptive. So here's the key stuff for adaptive. Adaptive is very specific. It goes after specific antigens, molecules on the surface of a pathogen. So it is said to be very specific. It has memory, meaning if you get a second exposure to the antigen, it can activate the immune response faster. It's not functional the moment you're born. It's not going to come in until you get a bunch of immunocompetent T cells. So it's going to come down the line, okay? So that's the compare and contrast of innate and adaptive. Now you've got some other immunity terminology here. Active, natural, passive, artificial. So how did we use that information? Here's my definition. Active. You are exposed to antigen. You have to go through the entire immune response. Passive. you're exposed to antibody. You do not go through the entire immune response. Natural or naturally acquired is what I say. Living your everyday life. Artificially or artificially acquired, as I say, medical procedure. Those are my definitions, but that's good enough. And so once upon a time, you had a lecture exam and you were given like 4 examples and you had to classify them based on, is that active, artificial, passive, natural, and write on your to-do list, go back into lecture exam 2, find those questions. You're not going to get the exact same questions. What would be the point? But use them to review the 4 examples and what falls into which category. There were a couple of questions in that exam too about comparing and contrasting innate and adaptive. Okay, and that'll be a good enough review. All right, I'm not going to go through this one because I want you to. And then if you're not comfortable, then you'll email or you'll stop by and you'll ask me to double check for you. But I want you to write this out. Be prepared to put the events of an immune response in order from when it is recognized with MHC, major histocompatibility proteins, to cell and antibody-mediated responses. Included in this are going to be things like macrophage, interleukin-1, presenting antigen, the CD4 helper T, blah, 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 blah. Okay, you're not going to get the same question, but in that same lecture exam too, there was an ordering question. So again, go through that, check out that question, and that'll prompt you because you'll now be able to see what the answer is. Again, you're not going to get the same question, but it'll at least remind you of what the steps of these events are. So put on your to-do list, make sure you can put the events of the immune response in their chronological order. Then after all of that, we hit the respiratory system. Okie dokie, so respiratory system, right? Well, one of the things we talked about in lab and in lectures actually are regions of that pharynx, like what's in there and what their functions are. And so you got 3, right? So you have nasopharynx here, oropharynx, laryngopharynx. Nasopharynx is airway. Okay, so that's how the air kind of enters in. And there's also a tonsil there, right? There's a pharyngeal tonsil up there. Now, once air goes past the nasopharynx, it goes to the back of your throat. That's basically oropharynx. Oropharynx is where food and air might mix together. And in your oropharynx, you still had tonsils. You have your lingual and palatine tonsils for immunity there. Now, once air goes past oropharynx and hits laryngopharynx, it gets kind of separated from the food with the epiglottis. And so once you get below the epiglottis in that laryngopharynx, you're back to having air only. And there's no immune system, there's no MALTs there. There's no mucosa-associated lympho-tissue there. In other words, there's no tonsils in the laryngopharynx. So be able to characterize what region are we dealing with and kind of what's in the region. Is it just air? Is it just food? Are food and air mixing together? Is there immune system there or not? That's it for that. Now we did also get pretty hardcore into the rest of the respiratory system, right? All right, pleura. That's the serous membrane that's sitting between your lungs and your ribs, and it happens to kind of be attached to both. Once upon a time in lecture, I was like, if you could pull your ribs up and out, you're going to pull your parietal pleura up and out, which means you're going to pull your visceral pleura up and out, which means you're going to pull your lungs up and out. It's all interconnected, right? And so the pleura, that fluid-filled serous membrane around each lung and attached to it, those are important. So again, there's a parietal layer up against the lungs, parietal layers up against rib cage. Visceral layer is attached to the lungs, and there's that cavity in between the two, right? And so that's the pleural cavity. That cavity contains serous fluid, and we talked about how important it is because you're going to have lungs expand and recoil under ribs. It's really important to have a slippery, no friction fluid barrier there. So as your lungs expand and recoil, they're not rubbing up against ribs or anything. there's a friction-free surface around them to move easily. Later on in the lecture, we talked about, back in the day, compliance and all the things that affect compliance. So like imagine not having a fluid-filled, slippery little bag outside each lung. Every time a lung touches something, that's going to be a problem, and that would decrease compliance. So that's the beauty of the pleura. We also talked a little bit about a pressure gradient associated with this cavity. A little too detailed for a final exam. The pressure gradient anyway. We talked a lot about alveoli. We said the respiratory membrane is extraordinarily thin, as thin as you can get in the human body and go between two organ systems. Part of it, of that respiratory membrane, is the simple squamous epithelia of the alveolus and its basement membrane. And that, yes, I know the dog's behind me. And that is bound, she's my mascot at this point, and that is bound to the basement membrane and simple squamous epithelia of the capillary. And so the capillaries wall and the alveoli wall are stuck together. So the respiratory membrane is two flat cells and a fused basement membrane. So super, super thin. And we know alveoli are blind in humans, so like it's just a little sac with one way in. So old air mixes with new air, so to speak. We know we have hundreds of millions of alveoli and a tremendous amount of surface area for gas exchange across the respiratory membrane. So again, this is kind of like the immune system. I'm not going to go through these events. I want you to, and then get back to me if you're confused. Pulmonary ventilation, that's breathing. So if you were to put the events in order, that's muscles, volumes, pressures, air. So for quiet or forced inhalation or exhalation, you need to be able to say, here are the muscles contracting, here are the two volumes changing, here are the two pressures and how they're changing. This is how air moves. Back in the day and to the moment of the final exam, breathing is muscles, volumes, pressures, then air, always in that order. External respiration, that's gas exchange between the alveolus and your blood supply. So when you write that process out or explain that process to somebody, you should be able to say things like high partial pressure of oxygen in the alveolus, low partial pressure of CO2 in the alveolus because you're breathing. Blood's coming to that alveolus will have a high PCO2 but a low PO2, and the actual movement of gases across the membrane is simple diffusion. Those gases follow their partial pressure gradients. Now, when you get to internal respiration, that's gas exchange between the tissues and the blood supply, and it's the opposite direction. So tissues, always metabolically active, so they're always using oxygen and making CO2. So the tissues have low PO2, high PCO2. The blood coming through them, if we were looking at a model in the lab, would be in a red painted vessel. So the blood coming to them, high PO2, low PCO2, and again, the gases are going to diffuse with their gradients. So pulmonary ventilation, breathing, muscle, volume, pressure, air. Be able to name the muscles, the volumes, the pressures, and describe how air moves. External respiration, simple diffusion because of the gas exchange. Be able to identify between alveolus and blood supply where the high PO2 and high PCO2 or low, where the gas gradients are. Same for internal respiration, gas exchange between tissues and blood supply. You should be able to diagram it out and say, okay, tissues have high PCO2 but low PO2. Blood is opposite, simple diffusion. Okay. Alternatively, if you were given the events, can you put them in their correct order? So make sure you're practicing that and ready for that. And again, if you're not sure if you've got the volumes or pressures correct, well, email me or stop by and we'll chat. Now, we talked about a lot of mechanisms in the body for regulating pulmonary ventilation because breathing not only gets you the oxygen you need to stay alive, it also gets rid of the CO2 and helps regulate blood pH. So CO2 levels and pH are a profound signal in the body for changing pulmonary ventilation rates. If CO2 levels in the interstitial fluid and blood rise, we become acidic. Make sure you review that chemical reaction. So again, if CO2 levels in blood and interstitial fluid rise, we become acidic. And we're going to need to perform hyperventilation. We need to breathe faster to get rid of the CO2 and go back to homeostasis. Conversely, if CO2 levels get a little too low, now our pH swings and we become too basic. And so we're going to end up having to do hypoventilation to slow things down and preserve what CO2 we have. So you want to be reviewing the level of CO2 in blood and fluid. How does that change your breathing rate? And you want to be able to review the terminology that goes with that. So here's 4 words for you. Ready? Write them down. Hit pause. Write them down. Hyperventilation, hypoventilation, hypercapnia, high CO2, hypocapnia, low CO2. So again, if you missed those, pause or rewind the video, stop by, ask me questions, email me your notes. This was one of those concepts that actually made a lot of sense in class when I was like, all right, you've been exercising, you're in the parking lot, you were late, you're like right into the testing center. And let's say, or you're running to work because you're late, and like your exercise, you got a lot of CO2 around, so should you breathe faster, breathe slower? And everyone's always like, I gotta breathe faster, right? And the reason you're breathing faster is to get rid of that extra CO2. So again, you should be able to explain to somebody the effect that CO2 levels in blood and fluid have on respiratory rate, and be prepared to use words like, get ready, hypercapnia, hypocapnia, hyperventilation, hypoventilation. Now after respiratory system, we then moved on to digestive system. This is just me using a cartoon to be funny. All right, so some digestive system things. Hepatic portal system was a big fat stinking deal. We didn't spend tons of time on it in the lecture. You identified it in the lab, but I do mention it in the lecture. And the whole reason it's there is because your liver processes bloodborne nutrients. We actually spent a lot of time on that, right? We had conversations about fat in the diet and fatty acid types and how that affects LDL and HDL and how sugar affects your triglyceride and it, right? And I was like, I'm not even getting into all the liver functions. So your liver is a metabolic organ. You have to send blood straight from spleen, stomach, small intestine, large intestine, pancreas too. You've got to sneak that blood over to the liver so the liver can process all those bloodborne nutrients and fill in any gaps if it's able to. Or reprocess and retransport the lipids for you that came in on lacteals and things like that, okay? So that's the role of the hepatic portal system. It's bringing blood from digestive system organs to the liver so the liver can process the bloodborne nutrients. Now again, you should be able to do this. So I'm not going to do it for you. I want you to be able to do it. So you want to be able to list and describe processes of digestion. So intake, be able to explain what that is and name a few body parts associated with intake. If intake doesn't sound familiar, another way of saying intake is eat. All right, breakdown. There's different breakdowns. You can do mechanical breakdown by making molecules smaller, or excuse me, particles smaller. You can do chemical breakdown by making molecules smaller. So I'm going to repeat breakdown. 2 forms of breakdown. If you smash your turkey sandwich, that's mechanical digestion. Mechanical digestion, a form of breakdown, makes particles smaller. So your surface area goes up and you can swallow without choking. Chemical digestion, a different form of breakdown, uses enzymes to break molecules apart. So be able to explain the two and give a couple of examples of each. All right, propulsion. Propulsion just means to move. And that's going to include things like peristalsis, segmentation. Yeah. All right. Absorption. Absorption is what happens in your jejunum to ileum and colon, right? Absorbing means to move something from the lumen into the blood supply. You should be able to explain the kinds of things that are being absorbed. And again, what are the basic organs that are doing the absorbing? Elimination is to get rid of waste. So you should be able to identify the organ that's eliminating and basics on how it does it. So you see in parentheses here in a few words. In a few words or less, can you describe what this process is and some body parts that take part in it? Some examples. Now with chemical digestion, we're going to go a little bit more detail because we spent a lot of time in lecture on that. Chemical digestion of these three macronutrients start in different places. Chemical digestion of carbohydrates starts in your mouth with salivary amylase. It will finish in your duodenum with pancreatic amylase. So mouth and small intestine are where you're going to perform chemical digestion of starch, and the primary enzyme is somebody's amylase. Chemical digestion of proteins. It starts in the stomach with pepsin and the acid that denatures the proteins. It's going to finish with different protease enzymes in your small intestine. Once upon a time, we named some of them like trypsin and chymotrypsin. So again, proteins, we're going to start that in the stomach. We will finish it in the small intestine, and there's some enzymes for you. Chemical digestion of fat. It kind of sort of starts in the stomach. There's a little gastric lipase. You just don't get a lot of it in the stomach because you haven't emulsified yet. In the small intestine, we're going to finish chemical digestion of fats. And don't forget, we actually have to start with a form of mechanical breakdown called emulsification. So we use bile to make fat droplets smaller so that the enzyme lipase works better. So you want to be able to identify the macronutrient, the main enzyme category or enzymes that go with it, where its chemical digestion starts and where it ends. You also want to be able to explain saliva and the salivary reflex. So the salivary reflex, it's cerebrum and facial cranial nerves. So think about food, smell food, be hungry, hear me talk about food. Right? Cerebral cortex stimulation activates that reflex and you start to spit. Like I'm spitting in my mouth now because I'm talking about it. Now, what is saliva then? So saliva is a hypoosmotic secretion. It contains a lot of waste products, a lot of electrolytes and things like that. But it also contains, again, salivary amylase and proteins that help lubricate it. So what you're chewing on is called a bolus, right? And so by dumping saliva on the bolus, you do several things. Write these down. You start chemical digestion of starch. You lubricate the bolus, and you start to solubilize the bolus. You start to liquefy it. Makes it easier to swallow. Makes digestion later, especially chemical digestion, a little faster because you broke the surface area down. It allows you to taste things better because, again, you're solubilizing molecules in your mouth. Hopefully you wrote those down. Now, when we talked about digestion, we talked about 3 phases and how each phase activates the next set of organs. So see it, smell it, get hungry, talk about it, have your brain interact with food. That's cephalic phase. Cephalic phase activates saliva production, and it starts gastric movement and secretion. So cephalic phase activates salivary reflex and starts stomach activity. Then you chew and swallow. And when you put molecules in your stomach and stretch the walls of your stomach, you activate the gastric phase. The gastric phase reinforces stomach activity and stimulates the small intestines and colon. When your stomach is all done solubilizing that bolus and turning it into chyme, chyme will enter your small intestine. So you've got chemicals in the small intestine. That activates the intestinal phase. In the intestinal phase, we switch activity of the stomach to peristalsis, and we reinforce activity in small intestine and colon. So you want to be able to identify those three phases, and again, give an overview. So maybe rewind and write down what I just said, just saying. An overview of what's happening in each of those three phases. Now, CCK, cholecystokinin, secretin, long reflexes, short reflexes, facial nerve, glossopharyngeal nerve, local reflex, gastric gastrin, intestinal gastrin, a little too detailed for a final exam. Now, we did lots of times, lecture and lab, go through the 4 tunics of the alimentary canal. We won't do it again. So you want to be able to briefly explain them and like what function is taking place in them. So mucosa, innermost lining in contact with the lumen, that's where absorption and secretion take place. Submucosa, primarily connective tissue. The submucosa is what houses the nervous system here, the plexes, the enteric and myenteric plexus. It's housing those nerve endings and big blood vessels. Muscularis externa, that's the movement one. So that's peristalsis, segmentation, churning. Serosa or adventitia, believe it or not, as skinny as they are, there's a function, and especially for serosa, it reduces friction. So again, you want to be able to put them in order, identify each one, be able to say things like serosa, it's the serous membrane, it reduces friction. Muscularis externa is smooth muscle. Submucosa is connective tissue that houses nerve endings and big blood vessels. Mucosa, well, it's epithelium and other stuff, and mucosa absorbs and secretes. All right, we are getting to the end, people. So after digestive system, then we got into urinary system. The urinary system questions have a higher point value because remember, this final exam is 70% urinary reproductive, 30% old stuff. And the reason there's a slightly higher point value on the urinaries is, you have more repro questions than you have urinary questions. So you can use the study guide, the final exam study guide for the urinary stuff to narrow it down. Make sure you're using reproductive system, learning objectives, the detailed stuff for that exam. All right, you should be able to trace something through the nephron, identify each part of the nephron, basic overall tissue type, basic overall function. So, glomerular capsule, simple squamous, filtration. Proximal convoluted tubule, simple cuboidal, reabsorb, secrete organic-based molecules. Loop of Henle reabsorbs water by osmosis. Ascending limb is pumping the salt. Descending limb is allowing the water to move. I think in the lecture I say ascending limb makes the gradient, descending limb uses the gradient, vasa recta capillary maintains the gradient. All right, distal convoluted tubule, simple cuboidal, reabsorbs, secretes electrolytes. Collecting duct, many nephrons dump into a collecting duct. Simple cuboidal, reabsorbs or secretes sodium under hormonal control. Aldosterone, ADH, AMP, just saying. Okay, so again, one of those things where I'm not going to give it to you in this video. I want you to write it out and get back to me. So if I gave you blood vessels and structures, would you be able to successfully trace through them? Like blood flow in and out, or reabsorption of a nutrient molecule, or say excretion of a waste molecule, kind of like lab. Be prepared for that. Something's going to happen with that. Okay, now with urine production, we talked a lot about control mechanisms. So we were like, there's intrinsic control, right? That's the juxtaglomerular apparatus and tubuloglomerular feedback and myogenic. That's just the afferent arteriole controlling itself or perikin's controlling it. We talked about sympathetic nervous system. Sympathetic nervous system works the same way in every single blood vessel in your body. If you fire off some sympathetic nervous system action potentials and you do it to this afferent arteriole, you're going to constrict it, which decreases GBP, decreases NFP, and decreases GFR. If you stop, then the reverse, like if you're parasympathetic mode, then the reverse starts to happen. So you want to be able to explain, okay, here's how we control filtration, right? We control it by way of afferent arteriole. Sometimes it can do it itself, that's autoregulation. Sometimes we can override with sympathetic nervous system. You also want to be able to describe, characterize, explain the process of filtration. So in filtration, remember, you've got 3 pressures. There is a glomerular blood pressure or glomerular hydrostatic pressure. That is the function of the amount of blood in the glomerulus, and it is a force pushing out. That is the only one that can be easily changed. So if you change your glomerular blood pressure, the pressure pushing out, you'll change the pressure gradient, net filtration pressure. And if you change the pressure gradient or the net filtration pressure, you will change the speed in which you filter or the glomerular filtration rate. Okay, so there's one pressure pushing out that we can change, and we do constantly change it. There are 2 pressures pushing back in. I'm just going to write keywords. One is a colloid, other keywords, osmotic. Okay. It's water molecules from the glomerular space trying to go back into the plasma because there's plasma proteins. It's the osmotic force. The other one is a hydrostatic pressure. Pretend I didn't write sloppily on my screen here. Hydrostatic pressure is in the same direction. It's the fact that the water molecules in the glomerular space that are already there are also just sticking to the outside of the glomerulus and pushing against it. So 3 pressures, pressure pushing out, glomerular blood pressure, 2 pressures pushing back in. One is an osmotic, the other is hydrostatic. The hydrostatic and the osmotic are not changed moment to moment. Osmotic only changes, remember, severe starvation, liver failure, kidney failure. That's kind of what would also cause the hydrostatic to change. So the only thing that changes under normal pressure is the glomerular one. So again, you could explain these 3 pressures. You could explain how changing glomerular hydrostatic pressure will result in changes in NFP and GFR and why it's important. So guess what? That's the old stuff. So what I want you to do now is go grab the original learning objectives, like the hardcore individual detailed ones for the respiratory system, and start writing those out. If you want me to double check them, I totally will. That's the end of this video. If you want a video for reproductive system, I totally got your back. It's already there. It's your online lecture. Okay? So you have an online lecture video and a bunch of resources on Blackboard to help you master reproductive system. So my lovelies, I wish you a fabulous day. I wish you a healthy day. I hope that it is filled with amazing awesomeness. It has been my distinct honor and my distinct pleasure to get to hang out these last 4 months with you with all things human body for the most part. If anything, I hope you learned a few critical things like do not use 28 days as a birth control method. Diabetes mellitus sucks. Don't get it. If you got it or you end up with it, manage it well. It's serious. Okay? Love each other. Love other people. Be kind, gracious, respectful, and nice in all times, because that's the way we roll at Prince George's Community College. All right, if you got any questions as you're studying for your lecture final exam, y'all know my door is open. Sometimes it's a virtual door, sometimes it's a door. Just send me an email first, and I am happy to go over any of this with you. Or find some cool video of somebody else's if you need it. So take care of yourself, be well, be gracious and wonderful, revel in the amazingness of you, and I will talk to you soon.